Hello and welcome back. We are going to be discussing the last chapter in our cardiovascular system unit, which is chapter 20. That is going to include the blood vessels. Some objectives that we are going to discuss in this chapter are going to be the blood vessels that actually make up the cardiovascular system, that are going to include the arteries, the veins, and the capillaries. We're going to discuss how blood flows to and from the heart, and those special sense structures that we touched upon in the heart chapter that are going to maintain or monitor the blood gas level as well as the blood pressure level. We are then going to get into the hemodynamics, or how blood actually flows, and how that's going to affect blood pressure, and how other things are going to affect how blood flows throughout the body. And then we will discuss the vasomotor regulation, which is going to entail the vasoconstriction and vasodilation of vessels. And then we will discuss how filtration and reabsorption occurs within the capillaries, and then eventually how blood is then going to return back to the heart. Finally, we will then discuss any clinical abnormalities or diagnosis that can be found within the blood vessels and how they actually function. So if we look at some histology slides of blood vessels, we're going to find that they are made up of three layers of tissue: the tunica externa, tunica media, and then the tunica interna. The tunica externa is going to be the outer part of the vessel wall. The middle part is going to be made up of smooth muscle. So we've got connective tissue on the externa, smooth muscle on the interna. So this is going to be an area where we're going to see that vasomotor regulation occurring, where constriction and dilation occur because of the muscle either relaxation or constriction. And then finally, internally, we're going to see the tunica interna. Now, this is made up of that simple squamous epithelial tissue that is going to be continuous with that endocardium that we saw within the heart. Now, these are going to secrete that cisplatin, which is going to be another chemical that's going to help propel those platelets so clotting does not actually occur. Remember, we talked about prostacyclin, which is going to be another chemical that's going to help propel or help remove any of those unwanted clots. So these layers are going to be consistent throughout the blood vessels within the arteries, veins, and the capillaries. However, we're going to see that they're going to look a little different in the arteries, in the veins, and in the capillaries. So starting with the arteries, we have three different types of arteries: conducting, distributing, and resistance arteries. Now, if you remember what we had spoke about in the heart, how the cardiovascular system is pretty much the heart is the motor, then the blood vessels are going to be those roads, and then the blood is going to be the transportation or the cars that are going to flow through those blood vessels. Now, much like roads that we talked about before, we're going to see that they're going to have different sizes. So these conducting arteries are going to be like the largest vessels. So they're going to be the aorta that extends off of the heart, and they are going to be the largest part of the arterial system. The next, the distributing arteries, as we move further away from the heart, we're going to find that the vessels are going to get smaller and smaller and smaller. So the distributing arteries are going to be much like those exit ramps off of the 405 that are going to take us to other means of the city. And then the resistance arteries are going to be the smallest ones, where we're going to see that those are going to be our residential roads. So those are going to eventually get into potentially dirt roads, which are going to extend like the capillary system. Now, looking at these three, we're going to see that the three walls that we just discussed are going to look a little different. The conducting arteries, again being the largest, are going to have to withstand a really great amount of pressure because they are maintaining or dealing with the pressure that is being conducted from that left ventricle contraction. So much like the Ripple system, we're going to see that as that left ventricle contracts, or the ventricles contract of the heart, it's going to perform that ripple system down the vessel walls. So the conducting arteries must be very large. Because of that, their vessel walls are going to be very thick. So the tunica externa is going to be very thick. The middle portion where we have a lot of smooth muscle, the media, tunica media, is going to be very thick. As we move down into the distributing arteries, we're going to find that the external part, or the externa, is going to start giving away, or it's going to start being reduced. Then the media is also going to start being reduced. Finally, into that third resistance arteries, where we're going to see that the tunica externa is very, very little, as well as the tunica media. Now, we need to do that because as we get to the capillaries, we're going to find that the tunica externa and the tunica media are completely gone. The reason we need this to occur is because remember what happens in the capillaries. We want gas exchange to occur. Well, gas is not going to flow to and from the tissue into the vessel and the vessel into the tissue if there is a concrete wall that's going to be hindering it. So as we move down, we're going to find that these walls are going to start minimizing. So again, these large conducting arteries are going to be the largest that are going to extend off of the heart, the pulmonary arteries, as well as the aortic trunks. These have to be larger, and they have to be more firm in their vessel walls because they are dealing with that expansion and recoil from that ventricular contraction or systole. Okay, so as the ventricles contract, we're going to find that the immediate vessels are going to have the highest pressure in there. So because of that high pressure, it needs to be maintained with these vessel walls. We're also going to see that they're very elastic, or they have a great amount of elasticity, meaning that they can expand and recoil on their own as well. Now, arteriosclerosis is a disease that happens when these vessel walls begin to stiffen. When they begin to stiffen, then they cannot expand and recoil or move like they should. Again, distributing and resistance arteries now are going to go from that large-sized vessel into this medium-sized, and these are going to be branches off of the aorta. So much like when we actually look at the vessels, we're going to find that they're going to be named after the location that they are going to feed. So if we look at the renal arteries, we're going to know that they're going to branch off of the aorta, and they're going to then extend into the kidneys and feed the kidney tissue. Resistance arteries then are going to be those even smaller arterioles that are going to, again, be our residential roads that are going to feed more tissue. And then finally into the capillaries. The capillaries at this point now only have that simple squamous endothelial layer as well as this basement membrane. So the tunica externa is completely gone and the tunica media is completely gone. Okay, this happens because, again, we need to get that gas exchange to occur. Now, if you recall, when we were looking at the blood chapter, we talked about the size and the diameter of a red blood cell, which is approximately 7.5 micrometers. And if we look at capillary size, we're going to look at the diameter of a capillary is about 5 micrometers. So if you remember when we talked about sickle cell anemia and how important the shape is for red blood cells, they need to be able to fit and maybe elongate, or they need to fold back on themselves to actually fit within these within the diameter of these particular capillaries. Okay, so we have 3 different types of capillaries that we are going to discuss, which are called the continuous capillaries, fenestrated capillaries, as well as the sinusoids. Now, the sinusoids are going to be a little bit different because they are going to be made up in liver tissue and the bone marrow, which we will discuss here shortly. Now, these are going to be able to be determined, or you'll be able to determine the type based on what is actually going to cross the membrane. Continuous capillaries are found throughout most tissue, but we're going to see that only small molecules are going to be allowed to be passing. So we're going to more or less see that there's more tight junctions that are going to help hold these cells together, as well as something called intercellular clefts. So these clefts are going to be very, very small pores that are going to allow these small solutes to pass through. So small molecules, much like glucose. However, these large proteins that we talked about before, these blood proteins, are not going to be able to pass. Whereas fenestrated capillaries are going to have filtration pores that are much bigger. So they're going to be about 20 to 100 nanometers wide. So they are going to allow the passage for small molecules, but they're not going to allow those larger proteins still to go through. However, we're going to see many things are going to be able to pass through much more frequently than the continuous capillaries. These are typically going to be found in organs, and we're going to find that they're going to allow for rapid absorption and filtration. So, which we'll talk about that when we get into that actual process. So we'll see these in the kidneys and the small intestine, because this is where we're going to have a great amount of nutrient absorption. The kidneys, we're going to find we're going to have solute reabsorption as well as water, and then the endocrine glands, obviously, because we are taking up those or releasing those hormones within the blood. Now, again, these sinusoid capillaries are going to pretty much have the largest gaps possible, and they're going to allow large proteins, blood cells, that albumin that we talked about, the blood proteins, those are going to be allowed to pass through very, very rapidly. So we're going to see that the spleen, liver, bone marrow, these are all going to have that spongy sinusoid tissue. Now, these capillary beds that we are going to discuss we're going to see how blood flow can potentially be regulated. It's going to be regulated by something called a precapillary sphincter, meaning that there is going to be a sphincter prior to allowing the blood into this capillary network, to where if the sphincter closes, we're going to see, again, remember, blood is going to flow from a higher pressure to a lower pressure. We're going to find that it can actually be regulated by constricting these.